Our topic is about antibabesial drugs. At the end of this discussion, we will be able to learn what is babesiosis and treatment for babesiosis. So what is babesiosis? A rare infectious disease caused by single-celled microorganisms or protozoa belonging to the Babesia family. Parasites that invade red blood cells or erythrocytes, usually carried and transmitted by ticks or vectors through its bite. Some direct animal-to-animal transmission may occur when an infected dog with oral lesions or abrasions bites another dog, characterized by fever, hemoglobinuria, anemia, and icterus. Eradication of the disease is based on control of vector ticks, chemotherapy, and in few countries, vaccination. Acridine derivatives. These drugs are effective against B. equi and used in horses as 5% solution. Systemically, these drugs are less toxic. Acriflavine, since 2014, has been undergoing testing as an anti-malarial drug to treat parasites with resistance to quinine and modern antiparasitic medicines. Euflavine, a topical disinfectant indicated in the treatment of wounds, blisters, insect bites, and infected wounds. Aromatic diamidines. These diamidine derivatives bind to DNA and interfere with parasitic replication. Diminazene produces its antiparasitic actions by selective blocking of kinetoplast DNA synthesis and replication, has a tendency to accumulate in tissues. Therefore, half-lives are very long, which may lead to residue problems in food-producing animals. Phenamidine acetonate. Phenamidine is an antiprotozoal drug of medicine, class used in veterinary medicine. It is used to treat Babesia infections, or babesiosis, dogs, horse, and cattle. Its mode of action is to intercalate nucleic acid, competitive inhibitor of arginine metabolism. Diminazene aceturate. Berinil, Ganaseg, and Veriben. Clinical uses. Antiprotozoal agent used to treat trypanosomiasis in dogs, sheep, goats, cattle, Babesia infections, hepatozoon, canis infections in dogs and horses, cytoxinosis in cats, leishmaniasis in humans. Its mode of action is to inhibit DNA synthesis in protozoa. It may not completely eradicate the organism, but because it is slowly metabolized, suppression of recurrence of clinical signs or prophylaxis can be attained for several weeks after a single dose. Carbanilides. A midocarb dipropionate acts by combining with nucleic acid of DNA in susceptible organism, causing the DNA to unwind and denature. Amicarbolide. It is active against all Babesia of cattle and horses. The quinolones inhibit bacterial enzymes, topoisomerases, including topoisomerase II and topoisomerase IV. Bacterial DNA supercoils and then uncoils during replication. Quinuronium sulphate, acaprine, baburan, pyruvan, and etc. These drugs have potent anticholesterase activity and have low margin of safety. The quinolones inhibit bacterial enzyme topoisomerases, including topoisomerase II and topoisomerase IV. Next is TRYPAN blue. Blocks C3b receptor on both erythrocyte membrane and parasite itself and probably prevents the parasite from entering the erythrocyte. It suppresses parasitemia and alleviates clinical signs but does not eliminate infection. Next is the toxicity of antibabesial drugs. All are potentially toxic. Immunocard is the least toxic while QUINURONIUM is the most toxic. Repeated use of diminazene and phenamidine causes degenerative change in liver, kidney, and heart. And that's all for antibabesial drugs. Thank you.